This video is going to show you how to use Punnett Squares as a graphic organizer to solve statistical problems in genetics. In this sample Punnett Square, you can see how outcomes can be similar to what you would do for flipping a coin. Flipping a coin, the probability of heads or tails is always 1/2. You can see that as written above each of the columns and rows in a Punnett Square, the 1/2 probability outcome. Just like in genetics, we would have alleles, and alleles would be a different form of a gene. There may be two forms, one coming from mom and one coming from dad, and we know through meiosis that you can only get one or the other because they'd be attached to chromosomes containing DNA. Now, as you can see here, the heads on the top left box come together, and you get a 1/4 chance of all the outcomes being heads. And that is, of course, 25% chance. If you look in the bottom right-hand corner, you will see that the outcome of having tails is 1/4. Another 25%. The probability of having heads and tails from the two coins being flipped is 50% because you have two boxes. It does not matter that heads is on top and tails is on top for those two boxes. They equal 1/2 probability, which is 50%. If you add up all the boxes, it should be 100% every time. 50% plus 25 is 75% plus 25% is our 100%. You'll see this is very similar to what we'll see in actual genetics problems. With this, you see our phenotype. It's the outward appearance, or the trait that you're looking for, is tails or heads. In genetics, it might be a color, it might be tallness or shortness, it could be some particular trait of human heredity. We have the ability to roll our tongue, as you can see in the picture. Not all people can actually roll their tongue. It's actually a dominant characteristic. Recessive characteristic is non-tongue rolling. We give genotypes to symbolize dominant and recessive. So big T is the ability to roll the tongue, and little t is the inability to roll the tongue. Anytime an individual inherits at least one big T, they will be able to roll their tongue. If an individual inherits both little t's, they will be unable to roll their tongue. This is where the role of dominance comes in play. If you have at least one dominant genotype, one dominant allele, it will overpower the recessive, and you will only see the dominant characteristic. In this example, you also see the gametes. Here is the female gametes, so these would be egg along this particular side of our Punnett Square. And then up here we have the male symbol, so these are the male gametes, so these would be the sperm, so put tails on those. You'll notice that both mom and dad are carriers for tongue rolling. And a carrier is also called a hybrid, because even though they have the characteristic in both these situations to roll their tongue, mom is a tongue roller and dad is a tongue roller, they carry the inability to roll their tongue as a hybrid. We call this a monohybrid cross. As you can see in the example, right, we have this sperm contributing a capital T and this egg contributing a capital T, and that gets us 25% chance of big T, but the phenotype, the outward trait, is actually going to be a tongue roller. If we take a look here, this egg and this sperm, right? Since this sperm has a lowercase t, the genotype's different. There's a 25% chance for this box to be a heterozygous hybrid. But the outward appearance for this individual's probability would be roller. Take a look here. This sperm and this egg produces another hybrid. They are a tongue roller. Another 25% chance. And the bottom egg in the last sperm produces our homozygous recessive individual, the non-roller. They're unable to roll their tongue. So when you look at this, we have 25%, 25%, 25%, 20%. That equals our 100%. And we can look at what is the genetic probabilities. All right. So there's two ways of looking at this. We want to know the phenotype probability and the genotype probability. So let's make it plural. For phenotypes, right, we can see that there's 1, 2, 3 rollers to 1 non-roller. So that's the typical phenotype ratio you get in a monohybrid cross. 3 to 1. It's 3 rollers to 1 non-roller. Genotypes is a little bit different. Genotypes in a monohybrid cross, we get a 1, a 2 to 1 ratio because you have 1 big T to 2 big T little t's. Remember, it doesn't matter which one came first in your Punnett square. The T always overpowers, so there's 2 of those, whether it's little t big T or big T little t, and there's only 1 non-roller, and that would be little t little t. Right, so in many problems you'll be asked for what is the phenotype probability and a genotype probability. You could always write this out in terms of percentages. So you can say 25, not 25, that'd be 75% rollers to 25% non-rollers, and be 25% big T, 50% big T, little t, and 25% little t. Just so it's the difference between a ratio and a percentage. This next problem helps us to look at how do you get hybrids in the first place. And this is the beginning of what we consider a monohybrid cross. A cross between a pea plant that is homozygous for yellow peas, which is capital Y, and a pea plant that is homozygous for green seeds, lowercase y, will produce only yellow heterozygous offspring. Whenever you're using these particular genotypes, you have to be careful because the capital Y's and lowercase y's could look very similar when you're writing them out. Alright, so we know that one parent is going to be capital Y, and the other parent is going to be lowercase y. Trying to show you some differences between them. So the only gametes that can be produced by this one parent is going to be the lowercase y, and they go on either side of our Punnett square. This parent here can only produce capital Y. When you plug it into our Punnett square, each box will get a capital Y and a lowercase y, meaning our probability is going to be 100% all hybrids. 100% all big Y, little y. Their phenotype will all be 100% yellow seeds. So you can see this would be a yellow phenotype, this would be yellow phenotype, this would be yellow phenotype, yellow phenotype. That's how you would figure out this is the beginning of a monohybrid cross. You wouldn't see the green until you get to the next generation. So if this is the parent generation, these are all your F1, your first filial generation. When you get to the F2 generation, you will see our classical 3 to 1 phenotype ratio and genotype ratio 1, 2 to 1, like we saw in the previous problem. This concludes this video.